Hello guys, how are you? Today we will talk about the cell cycle, which is the interphase plus mitosis in details. So now we are looking at one cell here. Here we can see a centrosome. Here you can see the nucleus with the nuclear membrane, and inside it you could find the DNA, which is in the form of chromatin, right? How many pieces do we have? We have 46 pieces of chromatin DNA, of course, because we are talking about a human cell. So now what happens? We said that when the order is given to the cell to divide, it must copy its DNA, which is the main thing that happens during interphase. The DNA must be copied. So the DNA duplication process starts, and then the DNA starts to duplicate, and they form, as you know, these pieces of chromatin. They start to duplicate and they become like this, right? So this is a duplicated DNA. You can notice the centromere in between, and so that they divide and they duplicate and duplicate, so that the amount of DNA will, what happens to it? It will be doubled. Why? Because all this DNA, it's make a copy of itself. Why the cell is making a copy? Because one set of DNA will be given to one cell and the other one will be given to the second cell, to the other cell. So now the DNA is copied. We have a copied DNA. But now it's when the DNA is like this, which looks like the noodles, as we said before, like spaghetti, it's very difficult for the cell to just separate these two sets of DNA from each other. So that this DNA must be organized. The DNA must be condensed, must be, compact, in order to just deal with it easily and separate the two copies from each other. How does it happen? Actually, it happens in the first phase of mitosis, which is the prophase, so that this form of DNA, the chromatin, will transform into what? It will transform into chromosome. So what happens? For example, I will just pick one of from the 46 pieces of copied DNA. When they coil, when they will coil and condensed, they will become like this. So they will become chromosome. Okay, so this is the socks here will represent the chromosomes. Okay, so that now we call it a chromosome, which is two sister chromatids. They are exactly copy of each other, and they are here connected by the centromere, of course. So that we will see that in the prophase. What will happen in the prophase? All this DNA, what it will just transform into what? It will transform into chromosomes. So we will deal with this. We will just represent these socks as chromosomes. Okay. Here we can see the different forms of DNA. As you know, normally this is the DNA. It looks like this, right? And we call it a chromatin, okay, which is a loose DNA. During interphase, what happens? This DNA, this one thread of DNA, we have 46 of them, of course. What happens? It makes a copy. It makes a copy by the process of DNA replication or DNA duplication, so that this chromatin, now we have two of them. We have a duplicated DNA. So this is DNA and this is its copy, and they are connected here by the centromere. And then when the cell is ready to divide in a prophase, what happens? This DNA start to organize, start to be condensed, to be packaged, and then they become a chromosome, an organized DNA. So this is an organized DNA. This is a chromosome. This now it's called a chromatid. This is a chromatid. They are sister chromatids because they are exactly copy of each other. But now in this form, the DNA is organized, condensed, and it's ready to start with prophase. So now let's move on to mitosis, and in the first division, prophase.